Hello everyone. Am I audible? Can
Yes,
someone
ma'am.
let me know? Hello? Okay. So, you guys, if you have any questions or if the audio or video, anything is not available or if it's not visible, please turn on your mics and let me know over the mic because I won't be looking at the chat session once I go into the presentation mode. Okay? Can I get a yes from someone over the mic? Somebody can just turn on your mic and tell me.
Yes,
Anyone?
ma'am.
Yeah, thank you. Thank you so much. Yeah, so I hope my presentation is also visible. Is it visible?
Yes, ma'am.
Yeah, so we can start the session. So this is for week 3 NPTEL course for experimental biotechnology. It is by Professor Vishal Trivedi from IIT Guwahati and I am your TA Sumati Raviraj from University of Hyderabad. So if you have any other queries or you need to contact me, you can contact me over this email ID. So this is for the week 3 assignment. So we will be covering topics which are related to the questions given in the week 3 assignment. In any topic, if you want more clarity or if you need more details or if you need any source or study material, you can let me know. I can provide it to you. So coming to the first topic, it is regarding the regions in a gene. So gene expression involves transcription, that is formation of RNA from DNA, and then translation, that is protein from RNA. This is the basic dogma. So for transcription to start, certain proteins, which are called as transcription factors, they need to bind to specific sequences on the DNA. Yeah. So to bind to the specific sequences, they also need to find them first. So these transcription factors find and bind to particular regions in the DNA sequences, and such regions are called as promoter regions. So promoter is a specific stretch of DNA which is located upstream. Upstream and downstream are terms used during when you're speaking about regions in a gene. So upstream is before and downstream is after the main coding sequence. So promoter region is generally present upstream to the coding sequence of a gene. It contains short DNA motifs and it is recognized by proteins called as transcription factors. And it is also the point where RNA polymerase binds. So these promoter regions have certain elements which are present, which are common in all eukaryotes. These are Tata boxes and CAT box, and they have a GC rich sequence. So this is conserved and it is common in all the eukaryotic promoter region. Then transcription factors bind here and recruit the RNA polymerase and position it correctly to start the RNA synthesis. So that is why it is very important for the transcription factors to bind to the promoter region so that they can properly recruit the RNA pol, and the RNA pol will then start transcription from DNA to RNA. Then other than this, the gene also has exon regions and intron regions in case of eukaryotic genome. And an exon region is nothing but a coding sequence or a coding region of the DNA that gets transcribed during the transcription process and then it gets translated into a protein. So they do not control the initiation of transcription, that is purely by the promoter region. but exon is the main coding sequence. Then we also have intron regions in eukaryotes. So intron regions are non-coding sequences between exons. So a gene might have multiple exons like exon 1, 2, 3, and between these exons you might have intron sequences which are non-coding sequences. They are transcribed but they are spliced before the translation. That is, you will find it in the mRNA, but once your mRNA has to be translated to make proteins, then they will be spliced out because these are not coding sequences. So you do not need these sequences while translation. And they also do not have any direct role in initiation of transcription. So here you can see a picture showing all the regions of a eukaryotic gene. So you have a promoter region, and not only the elements which I spoke, you have proximal core regions, then you have UTR sequences. You can also have enhancers and silencers, which might enhance the transcription or silence the gene. So help in silencing the gene. So this entire sequence is called as regulatory sequence along with the enhancer and silencer. Only this region is the promoter region. Then we have our open reading frame, which is our gene. So we have a start sequence and then it goes on with introns and exons alternating, and then we have a stop codon. So after that, also we have a UTR region, which is untranslated region. So you also have 5 prime and 3 prime untranslated regions. So then we also have terminators and downstream to the gene. So this is your gene. This region, which is before the gene, is your upstream region, and this region after your gene is your downstream region. So downstream also you might have enhancers and silencers. So this is also your regulatory sequence. So this is in your DNA. Then once your promoter region has your transcription factors, which will then recruit your RNA pol, you will have transcription process being initiated. So then we have our pre-mRNA, which will have your exons, introns, etc. And then we have post-translational transcriptional modifications. So we get our mature mRNA. Just a second. Guys, guys, I'm recording a class. Can you just uh, lower the volume? Yeah, sorry. So from pre-mRNA, we have post-transcription modifications, and we get mature mRNA that is which does not have any intron sequences. So these introns are removed by splicing. So then you have a mature mRNA which has only exon sequences. Then from the mature mRNA, we start the process of translation, and then finally we get the protein of interest. So this is how it happens. So in the post transcription modifications, we also have addition of certain regions like 5 prime cap and poly A tail at the 3 prime end. This will help in further translation. So these are the major basic regions of a eukaryotic gene. So the next one is DPC, and how why do we use DPC and what is used in DPC. So the enzyme that DPC is used to inactivate is called as ribonuclease. So the reason behind this is DPC is a chemical that modifies histidine, lysine, and tyrosine residues in proteins, thereby inactivating the RNases. So, why do we need this? RNases are very stable and ubiquitous enzymes. So, when you need to use RNA for your experiments, you do not want any sort of contamination from RNases. So you need to eliminate RNase completely from your solution in which you have your RNA. So for this reason, For this reason, we need DPC water, which will eliminate the activity of RNases. So we use this in lab, DPC treated water, usually 0.1% DPC. It is incubated overnight and then autoclaved. And this is a standard for preparing RNase free solutions in all the molecular biology experiments which involve RNA. So autoclaving removes the residual DPC, which can also be sometimes toxic and it can interfere with downstream reactions. So once DPC has done its job, which uh, we have incubated overnight, we can then autoclave it to remove the residual so that it doesn't interfere in further processing. So here we can see that other than DPC, there is also ultrafiltration where you can do so that you can eliminate any possible RNases. So you can see here the RNA in DPC treatment. You have clear two bands of 23S RNA and 16S RNA. So in ultrafiltration also the RNases are removed. So here also you have neat clear bands of RNA present. But in solution which was not treated with DPC, which could have been contaminated with RNases, or if you add RNase and check that if your RNA is present or not, you will not have any band for RNA because your RNAs have degraded the RNA. So it's very important to use DPC water or RNase free water, whenever you're using any solutions or making any experiments which involves preparing RNA samples or isolating RNA samples. So this has many applications. So inactivation of RNases, primary use is it prevents RNA degradation in the experiments, and it is essential for RNA related work or RNA related experiments like RT-PCR, northern blotting, or when you have to send your RNA for sequencing, etc. So this is very crucial. Then, other than this, preparation of RNase free water and solutions. Uh, as I said, 0.1% DPC is used. So, this is used to dissolve RNA samples, enzymes, and other reagents as well. So, decontamination of the glassware and plasticware is crucial before this. Otherwise, your DPC solution can be used to rinse your labware also to remove any sort of RNA contamination. In case you're reusing any test tube or glassware or plasticware, if you do not have autoclaved or if you're not sure whether they are RNase free, you can also rinse it once with DPC solution, though, so that they become free of any sort of RNase contamination. Then maintaining RNA integrity in experiments. So this ensures that RNA is intact during extraction, purification, and electrophoresis. Then histidine modification studies. So DPC reacts with imidazole groups of histidine residues in proteins, and it is used in biochemical research to identify histidine's role in enzyme. This is one uh, other function of DPC water or DPC treated water. So this was about DPC. So if you guys have any doubts during the presentation also, you can turn on your mic and let me know. And I might check the chat box sometime, but sometimes I might forget it. So let me please turn on the mic and let me know. So I hope I was audible. I saw that my mic was off. Was I audible?
Yes, ma'am.
Okay. Okay. So please let me know. Do not hesitate if any time if the mic is on or if there is any issue. So the next is the next topic is regarding the supershift assay. So the correct reaction mixture for supershift assay. So it is a mixture of DNA transcription factor and antibody. So the super shift assay is an advanced version of EMSA, that is electrophoretic mobility shift assay. It is basically designed to confirm the identity of a protein that is often a transcription factor, may not necessarily be any protein of interest, yeah, but it's mostly for the transcription factors because they bind specifically to the DNA. But if you want to see interaction of protein with your DNA sequence or gene of interest, EMSA is generally used. So it binds to a particular DNA sequence, and in EMSA you can see that some protein binds to the DNA and it has slower migration, but you can't tell which protein unless you add a specific antibody. So in case of super shift, this is your step. So that is where your mixture comes into play. You have your DNA, your transcription factor, or your protein, interacting protein, and then you have an antibody. So the antibody will basically help you to identify which transcription factor or which protein is bound to the DNA, which has a slower migration or which has a shift now. So because now you have an extra addition other than your protein of interest on your DNA, the shift will be double. So your first after addition of antibody, you will see that the run or the level at which your DNA is now will be much slower than your DNA and transcription factor, which will be slower than your regular DNA, which is probe free. So here you can see in the image, this is without any probe. So you have all of them at this point or at this front, but here you have your uh, protein of interest and your specific competitor or your gene of interest or your DNA sequence of interest. So this is running slower than your only DNA sequence. So you can see this sequence and this sequence is same, but now it has bound to a protein. So the DNA which is not bound has run much faster than the one that is bound with the protein. So this is your shift. And then if you need to identify this, you have another reaction which was run, which in which there was antibody. So because now it is also bound to an antibody, antibody is also a protein, so it has two proteins attached to it now. So this will run even more slower than your single protein and DNA sequence. So this is called super shift because it's a double shift. So this assay helps you identify, not only know if your transcription factor or your protein of interest is bound to the DNA sequence, but also identify which particular transcription factor or protein is being bound. So why is the mixture DNA plus transcription factor plus antibody? So DNA provides the sequence for protein binding, and transcription factor is the protein whose binding you are trying to detect, and antibody binds to the transcription factor, increasing the complex size, leading to the super shift. So without all the three you can't detect the super shift band. So basically your correct reaction mixture must contain DNA of interest or the region in which your protein might bind, and your transcription factor or protein, and also antibody. So all these three together in a mixture will give you a supershift assay or a supershift band in your electrophoresis. So these are how they have been separated on your gel. So you can see here in the first lane you have only
Ma'am,
your
voice is
protein.
not audible, madam.
Just a second. Okay. Is it audible now? I don't know why the mic is getting turned.
Yes, ma'am.
Yeah, so
I'm
let
also
me know.
screen
Yes,
is
ma'am.
not
Now it's
present.
audible.
Yeah, yeah. So how much of this super shift assay was audible?
Starting from the very fast part, it was not audible.
Oh, then why didn't you guys tell me before? Okay.
We
See,
think
that's what I
we
said.
have
I
thought.
won't be checking this like very often. I will check in at like 10-15 minutes interval. So better turn on the mic and let me know. There is some issue. The mic is getting turned off. Okay. So
Ma'am,
just let
if you are
me.
presenting screen also, screen is not visible right now.
Screen was also not visible. Yeah, please do tell me such things. What about now?
Now it is
Yes,
visible.
ma'am.
Now it's visible.
Yes, ma'am.
Yeah, okay. Thank you. Thank you. So let me know if such issues come again later. Yeah, so we'll go again about the supershift assay. Or so I hope this was visible and audible, the DPC part.
Ma'am, now
Was
it
it?
is visible and audible.
Okay, so I'll just show you even this also. So this was the gene of interest.
Last slide it was been. Yes, ma'am.
Yeah, eukaryotic gene I was talking about. I hope this was there, all the regions. So I think from here there was an issue, right?
Yes, ma'am.
Yeah, okay. So yeah, so basically I was talking about the DPC, which helps in eliminating RNases by degrading it. And you can see in the gel, this was the DPC treated water in which you can see your undegraded RNA bands. And ultrafiltration is another process in which you can eliminate any contamination by RNases. So you can also see the bands here, very clear, very neat, without any degradation or any sort of smear. So in case of a solution which was not treated with DPC, or if it was contaminated with RNAs, you can see that there is no RNA. Even though you might have isolated or extracted your RNA, if you do not dissolve it or elute it in DPC water or RNase free water, this is what happens. Your RNA will get degraded. So, okay. So, in case of supershift assay, like you can see, the reaction mixture I was talking about is DNA, transcription factor, and antibody. So, I'll just explain in the gel here. So, you can see that this is your probe, or this is your. You can also have multiple layers in this. You can have a competitor, you can have a mutant, or anything. So this probe is basically to see if it has been running, and you have your competitor sequence of DNA and you have your non-competitor sequence. So you can see that your non-competitor sequence has not bound to your protein, but the region that is your specific region which is complementary to your protein of interest, or the transcription factor of that particular gene has bound to only those sequences. So you can see in the first lane over here, there they have added only the probe. So you can see the probe at the end, and in the second lane you can see the specific competitor that is specific to your protein. So you can see that it has bound, and then there is a shift. So your probe is also something which has a similar length or similar size of your competitors. So it does, but however it does not bind to any protein. So it will run at the same speed if your specific competitor sequence has not bound to your protein. So you can basically think this as the sequence which is not bound to the protein generally. So this is the distance it will cover on the gel. But once it is bound, it will run much slower. So you can see when your free probe is much faster than your protein and sequence complex. Then in the third lane, you can see you have your probe, you have your specific competitor, and your protein. So here also you can see that it has been bound. Then in the fourth lane, you can see that you have your probe and you have your mutant competitor, which is not supposed to bind to your protein and your protein. So you can see that your mutant competitor has gone with your unbound probe, and your probe and your protein is present here. And in the fifth lane, you can see this is where the super shift comes. So here you have your antibody along with your specific competitor and protein of interest. So your protein of interest is bound to your specific competitor DNA sequence, and then you have your antibody bound to your protein. So your antibody is complementary, or it is against your protein of interest. So if it is, for example, beta actin, so your antibody of interest is anti beta actin antibody. So that will bind only to the beta actin protein. So now that it has bound, you can identify it because you have the antibody of interest specific to that particular protein. So this now will travel much slower than the previous only protein and specific competitor. So this is your shift, this is your super shift. So this is where your antibody also shows uh, slower migration because that is also protein. So this helps you identify which protein has interacted with your specific DNA sequence. So these were your, so yeah, so these are now the steps for it. So we prepare and label the DNA probe first. Just check. Yeah. So first is to prepare the labeled DNA probe. So this DNA contains the specific binding site for the transcription factor you are studying. Then it is usually labeled radioactively. So the star which you saw here was because it is labeled radioactively with mostly P32. So, or you can also use a non-radioactive tag like biotech. Then second step is to add the transcription factor, that is from purified protein or nuclear extract. Then transcription factor binds to the DNA probe, forming a DNA protein complex. In ENSA, this complex runs slower in a polyacrylamide gel than free DNA. And next is to add your specific antibody. So this will give you the super shift step. So the antibody binds to the transcription factor while it is bound to the DNA. So this creates a larger DNA protein antibody complex which will travel much slower. So then when you run it on a gel, the super shifted complex migrates even more slowly than the normal DNA protein complex. This shift confirms that the specific protein recognized by the antibody is indeed bound to the DNA. Okay. Then applications of this is to confirm the identity of DNA binding proteins in transcription regulation, then studying protein DNA interactions in promoter regions, then to detect binding specificity using mutant or competitor DNA probes, then verifying complex formations in nuclear extracts. This was about the supershift assay. Then coming to the next topic, it is regarding the tracking dye that is used in gel electrophoresis. So the most common tracking dye that is used is, it involves bromophenol blue. So bromophenol blue is a common tracking dye used in gel electrophoresis, including supershift assay to monitor the progress of the gel. So other stains like Comasi, even Stripan are for different purposes. Like Comasi blue is to stain the proteins after your gel is run. We would have discussed about this in the previous session. So once you have run your gel and then you want to visualize your protein bands, then you use Comasi blue. So it is not a tracking dye; it is to stain the proteins. Tracking dye is something which will help you monitor how far your lowest molecular weight or how far is your sample uh, running along with time, so that you can identify at which particular time you can stop the run and visualize your protein or go for the next step. So Evans blue, while Evans blue can be used in some biological experiment, it is not commonly used as tracking dye. It is more often used to study vascular permeability in retinal surgery or in retinal surgery. Then trypan blue was one of the options. So trypan blue is a dye which is used to check cell viability, not for protein or not for tracking. So trypan blue is a dye which we use to see if a cell is still alive or if we have any dead cells. So we use while cell counting during uh, cell culture or in vitro when we grow cells and when we have to passage and seed particular number of cells. So at that particular time, we need to know that how many live cells are there. So we use trypan blue for that, and it is not a tracking dye. So basically, the loading dye, whether it's for DNA or protein, they essentially have these coloring dyes which help understand which molecular weight or at what level your protein is in your gel in a particular time. So generally, for example, for DNA loading dye, we add bromophenol blue. Other than bromophenol blue, we can also add xylene cyanol and orange G. All these three are sometimes added together also because the three colors migrate at different rate and different levels. So you can see the lowest molecular weight and a higher molecular weight and differentiate at which particular time you can visualize it. So you can add one or more dyes. One dye most commonly used is bromophenol blue. Other than this, additionally, you can also add xylene cyanol and orangey, and these are the tracking dyes. Then we also have uh, high density reagents like glycerol, sucrose, or fecol that will be also added to your sample so that it gives your sample density. So when you release it into your well, it will go and nicely sit in the bottom of the well. Otherwise, it will get diffused in the buffer and you will lose your sample. And other than these two chemicals, we also have tris, SDS, EDTA, etc. So depending upon your need, you can add SDS or you don't have to add SDS if it's a DNA loading dye. But in case of proteins, you add SDS if it's for denaturing electrophoresis. If it is a native gel or a native page, then you won't be adding SDS. So this you can see a gel picture here. So in this they have all the three components. They have orange G, which is approximately 50 base pairs and runs approximately at 50 base pairs. Then we have bromophenol blue, which runs approximately around 300 to 500 base pairs. And then we have xylene cyanol, which runs around at 4,000 base pairs. So why a tracking dye is needed? So in native page or in EMSA or in any sort of gel electrophoresis, uh, particularly in case of DNA protein, and even if you want to see DNA protein complexes, they are all invisible while running. So unless and until you add a coloring component like a dye, you cannot visualize your sample running in the gel, and you would not know whether your sample has completed its run in the gel or whether it has released from your gel outside. So you have to have a color so that you can visualize it and stop your gel at your time of interest. So we need to know how far the gel has run to avoid running the samples off the gel. This is very basic. And excuse me, the tracking dye runs independently of DNA protein complex, giving a visual reference during the electrophoresis. So your tracking dye does not interact with your protein or your DNA or your sample. It is just present in your sample along with your protein or DNA. It will also run in the same rate along with the sample. So it is just to give you a line of visualization. Other than that, it does not react or it does not uh, interfere with your protein or DNA, especially in case of EMSA or anything. It does not involve or interact with your protein DNA complex. So why bromophenols blue? So like I said, bromophenols blue is used like very commonly in all the dyes. So because bromophenols blue is a small molecule and it is negatively charged at the pH in which we run the electrophoresis, and it migrates ahead of most of the DNA or DNA protein complexes in polyacrylamide gels, and it does not interact with DNA or protein. So we have all like plus 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 here. It's a small molecule. It's negatively charged. It migrates faster than your DNA protein. So you don't lose your sample. So it will run faster. So as soon as your bromogenol blue reaches the end of your gel, or if it's released also, you will be sure that your DNA or your DNA protein is still present in your gel. So another great point is that it does not interact with DNA or protein. So it won't interfere with binding or mobility, which is very important in case of EMSA. And of course, it is visible to the naked eye. It's a bright blue color that makes it easy for tracking. Then the migratory behavior. In case of polyacrylamide, it migrates around 10 to 15 base pairs size, and in case of agarose, it migrates around 500 base pairs size. So depending on the type of gel you're using also, and also the percentage of gel you're using, it will run faster or slower. So that, but it is relative to however your protein or DNA is also running. So you can easily use these, make use of this to know where your particular protein is still present in the gel so that you can further process it. So this was about bromophenol blue. So let me just check if everything's fine. Yeah, okay. So next is autoradiography. So autoradiography and DNA labeling with P32. So autoradiography is a technique that uses radioactive isotopes, and the radioactive isotopes helps to detect and visualize the biomolecules. So the radioactive emission exposed to photographic film or phospho imager screen, producing an image corresponding to the location of the labeled molecules. So basically, your uh, radioactive molecule that is present on the film or your sheet will be exposed to your photographic film or a phosphoimager, which will then lead to radioactive emission, and then it will show particularly where your labeled molecule is present. So by P32 for DNA. So DNA naturally contains phosphorus atoms. So it has a phosphate backbone. So it has phosphorus in it. So you can easily incorporate P32 in place of the normal non-radioactive phosphorus. So P32 is a radioactive isotope of phosphorus that can be incorporated into the DNA during synthesis or by labeling. Then it emits high energy beta particles, uh, which strongly expose the film, and it produces intense signal which can be detected to locate the specific location of your molecule. Then shorter exposure times are needed compared to lower energy isotopes. So this is one of the advantages of using P32. Then the process involves label the DNA with P32 using kinase reactions or Nick translation or PCR incorporation, and then separate the DNA fragments via your gel electrophoresis. Place the gel against the extra X-ray film in a cassette. Then beta particles from your P32 expose the film and develop the film to visualize the DNA bands. So this is your basic protocol. Then why P32 when compared to other isotopes? So we can also use tritium, that is your hydrogen isotope, and carbon 14. You know it is used in carbon dating, but they emit low beta particles. So the pros for these are they are high spatial resolution. But when you use these, you have more resolution for cellular localization studies, but it requires very long exposure times for DNA detection. So P32 is preferred for DNA blotting, sequencing, footprinting, hybridization assays because it has a stronger signal. Then your basic applications for your this is in southern northern blot hybridization. You could use it for DNA sequencing, Maxim, Gilbert, Sanger, labeled with primers. Then DNA is one footprinting, then gel shift assays, mapping transcription factor, binding sites, etc. Then excuse me. So this is a figure you can see for autoradiography. This is your gel is kept and a film is kept, and then you have your x-ray and it will be exposed, and then your film will show wherever your radioactive labeled DNA bands are present, it will give bands in this way. So, a gel containing radioactive DNA is dried and a piece of photographic film is laid over the top. The two are loaded into cassette case that prevents light from entering. After some time, the film is developed and dark lines appear where your radioactive DNA was present. So you can basically locate your DNA on this film because it has been radioactively labeled with your phosphorus isotope. So this was about your autoradiography. Next is visualizing your DNA protein complexes. So the best electrophoretic technique for visualizing DNA protein complexes is a non-denaturing gel electrophoresis. So a non-denaturing gel electrophoresis is preferred because it preserves the native structure because it's non-denaturing. You do not have any sorts of heat treatment. You do not have your detergent, so your protein or the protein is in its native form. So the native structure of both your DNA and your protein is present in this, allowing them to interact and form a complex that can be visualized. So in the previous sessions, we also discussed that one of the applications of native gels is to see interactions in the native forms, and most of the interactions you can see only when the protein or the DNA are in their native structures. So if you were to use a denaturing gel, the protein would unfold and the DNA protein complex would be disrupted, making it impossible to visualize the complex. So this was to visualize your DNA protein complex. The other options which were given were pulse field gel electrophoresis, denaturing gel electrophoresis, and capillary electrophoresis. So, a pulse field is a technique to separate large DNA molecules, and it does not involve any DNA protein complexes. Um, Denaturing gel, like we discussed, this technique disrupts the native structure of the proteins and nucleic acids, making it unsuitable for visualizing complexes. Then capillary electrophoresis. So this is a technique that uses capillaries to separate molecules, but it does not specifically uh, help in visualizing any sort of protein DNA complexes. So the right answer to this is your non-denaturing gel electrophoresis. So the basic principle behind your non-denaturing gel electrophoresis is to use conditions that preserve the native conformation of the protein, and then it helps in also preserving the interactions which it might have, and this means that the protein's overall shape and charge as well as its interaction with other molecules like DNA, influence its migration through the gel. So basically, you have your ENSA, your shift assay, etc. Then gel matrix. So polyacrylamide gels are generally used, and with the pore size that has been adjusted based on the size of the protein DNA complex that is being analyzed. Then buffers, non-denaturing buffers that maintain the protein's native charge and prevent unfolding. Then, during electrophoresis, an electric field is applied and the protein DNA complexes migrate through the gel according to their size, shape, and charge. Visualizing the complexes. After electrophoresis, the complexes are visualized using staining methods like Kumasi blue or silver staining, or by using fluorescently labeled proteins or DNA. So like we have many ways to visualize the complexes. Like we said, we can do either tagging them, or fluorescently labeling them, adding antibodies to them, fluorescently labeled antibodies or tagged antibodies, or you can also generally just stain the gel to just see the shift, or silver stain them to see the complexes if they are in like very minute concentration, because silver staining is much more sensitive than Comasi blue. Then coming to its applications, we have our protein DNA interaction studies. So, non denaturing page is valuable for investigating how the proteins bind to the DNA, including identifying the stoichiometry, that is, the number of proteins bound to the DNA, and affinity of these interactions. So, more the number of proteins that are bound, more is the interaction, and then more is the uh, you know shift, etc. Then analyzing protein complexes. It helps us to determine the oligomeric state or oligomeric nature of the protein as well. So when you're trying to see whether a protein exists in monomeric form or in multimeric forms, then you need to run a native gel. If you have a denaturing gel, everything will be in monomeric single line. So when you analyze such kind of studies, or when you do such kind of studies where you need to find out the oligomeric state of the protein, then you use native gels or non-denaturing gels. So it can help determine the oligomeric state of the protein complexes, including those involving DNA binding partners. Then separating and characterizing biological nanoparticles. Non-denaturing gels can also be used to separate and analyze larger complexes like viruses and other biological nanoparticles. So your biological nanoparticles can also be separated or analyzed using non-denaturing gels, because it does not degrade or denature your proteins present in them. So the advantages of your non-denaturing gel is preserving native structure. This allows for the study of protein DNA interactions in a more biologically relevant context, can relieve can also reveal information about protein DNA complex structure, that is, the migration pattern, can provide insights into the size, shape, and overall conformation of the complex. Then it is versatile. It can be adapted for various protein sizes and can be modified for different applications and different design of experiments and different setups depending on the nature and your complexes. Then it also has some limitations because it is a native gel. The resolution is less than your SDS page. While it preserves the native structure, non-denaturing page may not provide as high resolution as SDS page for separating proteins of similar sizes. So sometimes, because it is not denatured, your similar size proteins not might not be able to differentiate between them. Like if the sizes are too close, then they might appear as a single band even though they are two different proteins. So this does not happen in case of denaturing gels like SDS. So this is one of the limitations. And The other is complex interpretation. So interpreting the migratory patterns can be more challenging than with denaturing gels, as the mobility is influenced here by multiple factors. So you have to have a lot of experience, you have to know a lot of concepts, you have to know uh, your proteins of interest you're running, and how and what type of analysis should be made at particular mobility and migration patterns, because it's not so simple like in case of denaturing, that is your SDS page, where it's like gets separated based on charge and size. Here many other factors also influence, like the oligomeric nature, its interaction with other proteins, its interaction with DNA, etc., etc. So your interpretation must be very, very appropriate accordingly to your complex. Then sensitivity. So the detection sensitivity can be a limiting factor, especially when analyzing low abundance complexes. So if you have less abundance of your complex, your detection might not be there because it's not very sensitive. So this is another limitation of your non-denaturing gel. So the next is unlabeled oligonucleotides in case of gel mobility shift assay. I hope I'm audible. Yeah. Anyone could tell me where I can get the slides and PDF? Yeah, I will be sharing the slides, and I will also be uploading the video on YouTube, Rosni. So it will reach you
Okay,
there.
ma'am. Thank you.
Yeah. So does anybody have any doubts up until now? Maybe we can take a 5 minute break. So, do you have any doubts in these questions till now? These topics? Did you understand
No,
everything?
ma'am.
If you want me to explain anything
Yes,
again,
ma'am.
okay, just let me know. I'll be back in 2 minutes. If you have any doubts, you can type it over here in the chat box. So, it is 7:02. I'll be back by 7:04. Yeah. So, does anybody have any doubt? Then we shall continue. So the next is in gel mobility shift assay. The reappearance of the labeled oligonucleotide will be visualized by addition of unlabeled oligonucleotides. So, gel mobility shift assay is basically to detect and study DNA protein interactions. Like we discussed for the transcription factors binding to DNA, we do gel mobility shift assay. It's also called as EMSA. Then, electropoietic mobility shift assay. So, the basic principle is a labeled nucleotide, usually labeled with P32, like we discussed, or a fluorescently tagged oligonucleotide, migrates at a certain speed in a non denaturing polyacrylamide gel. So, when a protein binds to this labeled DNA, the complex is bigger and moves more slowly in the gel, producing a shifted band. That is what we saw previously. And then we also had super shift by addition of antibody. So, like I said, this is also called as EMSA. The binding between a labeled oligonucleotide, that is your DNA probe, and a transcription factor causes the DNA to migrate more slowly in a non denaturing gel. If you add excess of the unlabeled oligonucleotide, which is also called as cold nucleotide, so your radioactively or fluorescently labeled DNA is called as hot uh, oligonucleotides, and your unlabeled ones are called as cold nucleotides. So if you add excess of your unlabeled oligonucleotides with the same sequence, it is very important to remember that they must have the same sequence. They will compete with the labeled probe for the transcription factor binding. So your transcription factor will now bind to your unlabeled because now they are in excess. So your labeled ones will get replaced. So once it gets replaced, or this competition displaces your labeled DNA, it leads to reappearance of your free labeled oligonucleotide in the gel. So basically, if you want your free labeled oligonucleotide to reappear in the gel, you have to just add unlabeled oligonucleotides of the same sequence. So this is called a competition assay, and it confirms that the shift is sequence specific. So this basically says that because your hot and cold both oligonucleotides is of the same sequence, it basically says that this particular sequence was bound to the transcription factor, and it was not some random or non-specific binding. So this is sort of a confirmation step. Adding unlabeled oligos in excess competes away the labeled probe. So therefore, reappearance of free probe is seen and interaction is specific. Then how it differs from a supershift assay. So like I said, it is uh, EMSA, but it is not supershift, because in supershift we add antibody. In case of supershift assays, you add an antibody specific to the protein after the protein DNA complex forms. That is to identify the specific protein of interest. And the antibody binds to the protein, making the complex even larger, so it moves even more slower, and it produces an extra shift than the original shift. That is why it is a super shift. So super shift basically confirms which protein is present, and your competition assay confirms which sequence is specifically binding. So your super shift is to confirm the specificity of the your protein, and your competition assay is to confirm the specificity of your sequence. That is basically identifying which protein in super shift and which sequence in case of your competition assay. So you can see here, this is your unbound DNA, this is your protein bound DNA, which is your first shift, and then your antibody bound DNA, which is your super shift. So this is your super shift. So this was about unlabeled use of unlabeled oligonucleotides in case of gel mobility assay. So next is the source organelle for transcription factors in DNA footprinting. So transcription factors are basically found in the nucleus. So the nucleus is the cell organelle that is the source of transcription factors. and which is used in footprinting assays. So, transcription factors or proteins that regulate the gene expression are synthesized in the cell's nucleus. So, you can see these are four major organelles of the cell. This is your nucleus, this is your mitochondria, this is your endoplasmic reticulum, and this is your Golgi apparatus. So, your nucleus is a membrane bound organelle and it contains genetic information in that is DNA within the chromosomes. Along with it, it is the source for your transcription factors. Whereas mitochondria is also a membrane bound organelle, but it is a source for chemical energy that is ATP. And endoplasmic reticulum is also a membrane bound organelle. There are two types, rough and smooth. So, rough is protein production and ribosomes, uh, the place where ribosomes are present. That is why it is called rough because it has ribosomes attached to it. And smooth is lipid or fat production and it does not have any ribosomes. In case of Golgi apparatus, it is also a membrane bound organelle, but it packages the proteins and lipids into vesicles for transport. So coming to the nucleus as the source for this, so transcription factors and that are proteins that regulate the gene expression are synthesized in the cell's nucleus. They bind to specific DNA sequences, like we discussed, which are then used in footprinting assays to identify where these proteins bind on the DNA. So you are basically finding out the region in the DNA where the transcription factors bind. So that is called footprinting, and the nucleus uh, also houses DNA where transcription takes place, further linking it to the source of transcription factors. So, like I discussed, Golgi is primarily involved in processing and packaging proteins and lipids for transport to other parts of the cell and is not involved in transcription or DNA binding. ER is involved in protein and lipid synthesis and transport, but not in transcription factor production. Vacuoles are storage sacs and they are not involved in transcription factor or protein synthesis pathways that generate any transcription factors. They are just storage sacs. So, coming to the footprinting assays. So, footprinting assays are used to determine the specific DNA sequence to which a transcription factor binds. So, here we also have something called as a control binding reaction. So, a control binding reaction in this context usually contains DNA and transcription factor. So, the specific DNA sequence that is suspected to be a binding site for transcription factor, and a transcription factor is basically the protein that is believed to bind to the DNA sequence. So, this is what is present in your control binding reaction. So, by incubating the DNA with the transcription factor under controlled conditions and then subjecting the mixture to nuclease digestion, we can identify the protected DNA fragments that represent the binding sites. So, therefore, both the DNA and transcription factor need to be present in the control reaction to assess this interaction. Why the control contains DNA plus transcription factor? So, in footprinting, you have two main reactions. You have your control reaction and your reference reaction. So, in your control reaction, you have your DNA and transcription factor, which shows the protection pattern due to binding. Then, in case of your reference, there is no protein, uh, DNA alone. So, this shows full digestion pattern with no protected region. So, when your protein is not bound to your DNA, all the regions that is present in it, after using your nuclease digestion, all the regions will be cleaved at all possible sites. So, you will have a full digested pattern. Like if you have a length of DNA and it has two digestion sites, and you use your digestion enzymes, so you'll get three bands. So, that is your full digestion pattern. But in case you have a protein that binds to it or a transcription factor that is bound to it, it might bind in a place uh, in which your site is also included mostly because your restriction site is very small. So when it is bound to the protein, your digestion nuclease cannot go and digest that region. So instead of getting 3 bands, you might get only 2 bands. So this shows that there is binding in this reaction. So if you left out the transcription factor in the control, you also see a full digestion, like in case of reference. So in both samples, meaning you can't distinguish the binding. So that is why you need your transcription factor in the control. So the key points about control binding reaction is DNA must be the exact fragment suspected to contain the binding site, protein purified transcription factor or nuclear extract containing it, and binding conditions should include correct salt concentrations, that is your buffers, along with your cofactors, proper pH, temperature, etc., to maintain the protein DNA interactions. So basically, your outcome is protected footprint, which appears only when your transcription factor is present. So, what are the applications for these? So, it helps in identifying the transcription factor binding sites at a single nucleotide resolution. So, the resolution is so good that even at like the difference between one single nucleotide binding can also be identified. Then map promoter and enhancer elements within the gene regulatory sequence. You can see or you can map your regions in your gene and determine DNA protein interaction specific for different sequences, specificity for different sequences, like if the same protein is binding to many sequences, which has more affinity or which DNA sequence it is specific for. Then comparing binding affinities between wild type and mutant transcription factors. If your mutant transcription factor has lost the ability to interact or it is unable to bind to it, etc. Then also to study the effects of DNA mutations on protein binding and investigate the epigenetic regulations such as how DNA methylation affects binding. Then validate results from other DNA protein interactions like EMSA or CHIP assays. So these are also other assays which we use. So this is like a confirmation to any other experiment that you might have done. Then to characterize novel DNA binding proteins and and the recognition sequences. Like if you have a new protein, you don't know whether it binds to the DNA or not. You can try it out in this, and eventually you can see if it is bound, your digestion pattern will change. Then small screen for small molecules or drugs that can disrupt DNA protein interaction. So this is also one interesting application that you can see in drug testing and small molecules if it interacts any sort of DNA and proteins complexes. Then examine differences in binding patterns under varying physiological conditions or stress. So basically, if you want to see in disease condition, if any sort of interaction has been disrupted, or like in mutations, any mutants, if the interactions are disrupted, such kind of experiments can also be done. So this was about your footprinting assay. So this is your basic flow. You have your genomic DNA, you amplify it with PCR because you need abundance, and then labeled template DNA containing your protein binding site. So you have basically labeled it now so that you can visualize it, and then you add your protein of interest, and after adding you can see your protein of interest has bound to its specific sites. So when there is no protein, you can see your restriction enzymes or your nuclease digestion will cut or cleave your DNA at this site for this site for this sequence, this site for this sequence, and this site for this sequence. But in case of bound form, that is when your protein is bound, you can see that here you can see a cut, here you can see a cut, here you can see a cut, and here you can see a cut. Other regions where the protein is bound, you cannot see a cut. So this will help you in identifying the regions where the interactions occur. So then when you put it in a form of a library or a footprint, then you will see the region which has been protected, which was not cleaved by the digestion. So this sort of footprinting is done. Then, just a second. Yeah. So then we have how to purify labeled oligonucleotides. So the most appropriate analytical technique from the given options to purify labeled oligonucleotides is ion exchange chromatography. So, ion exchange chromatography is particularly suitable for purifying charged molecules like oligonucleotides because it has the ability to selectively bind to the ions of the opposite charge on a stationary phase. This allows for efficient separation from impurities. Other options which were given like HPLC or TLC or HPTLC. While HPLC is a powerful analytical technique for separation and for analysis of complex mixture, it is not specifically designed for purifying oligonucleotides based on their charge. So it is more suitable for analyzing the composition and purity of the purified oligonucleotide uh, instead of using it during the purification process. Then TLC, which is thin layer chromatography. It is a qualitative technique used for identifying and monitoring the progress of reactions, not for large scale purification. It is relatively simple and inexpensive, but it also lacks the resolution and the capacity which is needed for purification of oligonucleotides. Then coming to HPTLC, that is high performance thin layer chromatography, it is an improved version of TLC with much better resolution, but still it is not suitable for large scale oligonucleotide purification because it has limitations in case of sample loading and stationary phase capacity. So the best option you can go for is ion exchange chromatography. So this is your basic flow. You have your polymer beads with negatively charged functional groups. Then you have your protein mixture, which is added into the column containing the cation exchanges. Then you have your proteins that move through the column at rates determined by their net charge at the pH that is being used. And with cation exchanges, proteins with more negative net charge move faster and elute earlier. So large, you can see like different mixture in this, like large net positive, net negative, large net negative. This mixture contains all of it. So it's separating it based on the charge. So you have your negative charge, that is then you have your positively charged proteins that bind to the negatively charged bead. Then you have your negatively charged proteins which just flows through because it does not bind to the negatively charged beads. So you also have cationic exchanges and anionic exchanges. So cationic exchanges possesses negatively charged groups and these will attract positively charged ions. That's why it's called cationic exchanges, because it exchanges the cations. So these exchanges are also called as acidic ion exchange materials because their negative charge results from the ionization of acidic groups. Then anionic exchanger is opposite to it. It has positively charged groups and it will attract negatively charged anions. And these are also called as basic ion exchange materials because they have basic groups. So this was about ion exchange. Then we have PMSF. So during nuclear extract preparation, PMSF, that is phenyl methyl sulfonyl fluoride, serves as a protease inhibitor. It prevents the degradation of target proteins. So your proteases are basically which degrade your proteins. So PMSF is an inhibitor of this protein so that your proteins don't get degraded. So the target proteins is by serine proteases. Specifically, PMSF irreversibly inhibits serine proteases by covalently modifying a serine residue on their active site. This helps in maintaining the integrity of the proteins of interest by preventing their breakdown during extraction process. So, what are serine proteases? Serine proteases are a class of enzymes that cleave peptide bonds in proteins. They are commonly found in cells and can degrade proteins. including those being isolated from nuclear extract. So if they are getting degraded, then you don't have anything left in your sample. So PMSF is a chemical compound that reacts with the active site of serine proteases, specifically targeting the serine residue, crucial for catalytic activity. So that's how it inhibits it. This reaction inactivates the enzyme, preventing it from breaking down other proteins. So you can see this is usually the process for nuclear extraction. So you have cells, you swell them and you in a hypertonic buffer, and then you homogenize them with the Downs homogenizer. Then you have salt extract nuclei, and then you have cytoplasm separated. Then you concentrate and dialyze it in small scale nuclear extract. Then you have your identifications, your tRNA, your snRNA, etc. Then your pre-RNA, mRNA, mRNA, exon, etc., etc., etc. in this. So for all your proteins that has to be present if required in your nuclear extract, you need to add PMSF also. Then why is it important for nuclear extract is nuclear extracts are often used in studying DNA protein interactions and other nuclear processes. The presence of active proteases in the extract can lead to degradation of these proteins, compromising the results in the study. So, PMSF is typically added to the lysis buffer immediately. Like when you harvest the cells and you start your process, it's immediately added to the lysis buffer or other solution during the nuclear extraction procedure, ideally just before or during the lysis. This ensures that it is present and active during the protein extraction process. Then the stability, PMSF is known to be unstable and has a short half-life. Therefore, it is important to add fresh PMSF at each step of extraction procedure when protein degradation is a concern. Other protein inhibitors, like while PMSF is widely used, it is also used in conjunction with other protease inhibitors such as protease inhibitor cocktail. So you have a cocktail, you have like tablets which we use, which has many protease inhibitors mixed together like a mixture, which provides a broader range protection against various proteases, because PMSF is specific to serine. So you might have proteases which are not serine proteases. So in such cases, we use inhibitor cocktails. Then coming to Lowry method. So this is a protein estimation method. So Lowry is a calorimetric assay used for estimating protein concentration in a sample. It's based on the reaction of copper ions with peptide bonds under alkaline conditions. So you have alkaline buffer and then you have a copper solution, followed by reduction of Folin cyclosho reagent by tyrosine and tryptophan residues in the protein. So this produces a blue color that can be measured spectrophotometrically. This method is known for its sensitivity, especially at lower protein concentrations, and it is widely used in biochemistry and molecular biology laboratories. And there are other methods also which are used for protein estimation, like your biuret and your BCA method. All of them more or less use the same principle. So your biuret reaction in alkaline environment, copper ions react with peptide bonds in the proteins, reducing them to cuprous ions. So Cu plus 2 becomes Cu plus when it reacts with the peptide bonds in the protein. So this gives your color. So your Pauling reaction, reagent containing phosphomolybdic phosphotungstic acid is reduced by the tyrosine and tryptophan residues in the protein. This forms a blue colored complex. So heteropolymolybdenum blue. The color measurement is then taken after you get your reaction. So the intensity of the blue color, which is directly proportional to the protein concentration. The more protein you have, the intensity of your color will increase. So it is measured spectrophotometrically at a particular wavelength for particular reagents. So for Lowry, it is usually done at 750 nanometers. So you have your standard protein here. This is your copper plus 2 ions. And when in alkaline conditions, you have your standard protein cuprous complex, it becomes U plus. Then you have your fallen shore calcium reagent, and this will convert the color to a brilliant blue color because it has uh, your phosphomolytic tungstic acid. So once you get the blue color, your reaction is completed. You have to immediately take the readings and you should plot a standard graph. So before uh, finding out the concentration of any unknown protein, you have to first serially dilute the concentration of a standard like BSA, etc., so that you have the concentrations, and then you find out their uh, absorbance. So this y-axis is your absorbance, and these are your known concentrations. Then you get a standard graph like this with a straight line. So you know if your concentration is zero point 0.2, your absorbance is also zero point 0.2 in this case. So if your concentration is zero point 0.4, your absorbance is maybe 0.25. So that is how you take. Then when you have an unknown sample of unknown concentration, you take your OD. So for example, in the unknown, your OD was zero point 0.6. Then you have to put it in your standard graph. So if it is zero point 0.6, you see your, if you draw a straight line for the standard line, and draw another straight line at the x-axis, you get zero point 0.8. So if your OD was zero point 0.6, according to your standard, your concentration will be zero point 0.8 mg per ml. So that is how you basically do your protein estimation. So the key steps in consideration is the reagents. The Lowry method involves several reagents including alkaline copper solution and pollen culture reagent, and pH, the reaction is highly sensitive to pH, and optimal pH range is typically between 10 to 10.5. Since we need alkaline environment, we definitely need 10 to 10.5 pH. So the incubation time with fallen Sure, reagent is crucial for color development and reproducibility. Then interferences, certain compounds like some amino acid derivatives, buffers, and other chemicals can interfere with the reaction and affect the accuracy of the protein estimation. So, for example, the buffer in which you have suspended your protein might also interfere with it. So what you do is you take a blank in which you add only the buffer without your protein. So in that case, you can just subtract any reading that you get from your buffer from your buffer protein solution, so that it will be like subtracting the background which is not required. Then sensitivity. The Lowry method is particularly sensitive for detecting low concentration of proteins, often in the range of 1 to 1000 micrograms per milliliter. Then modified Lowry is also there. There are variations of Lowry method, such as modified Lowry, are available to improve the sensitivity of the assay and reduce the interference of certain compounds. So advantages are high sensitivity, reproducibility, and disadvantages are sensitive to pH and interferences, and then it has multiple steps, and it is sometimes variable because the pollen shock HAL2 reagent can vary in its reactivity, leading to potential inconsistencies. So like I said, there are other assays as well, biuret, and BSA. All of them involve the copper ions. Here, in case of Lowry, you use the fallen Schockel reagent, and in biuret, you use the biuret reagent, and in case of BCA, you use your bisynchronic acid reagent. So the reagents differ and the names differ according to the reagents. That's it. And if you want to know more about your biuret and BCA, you can watch the YouTube video. I have pasted a link here. And next is the chemical used to denature secondary structure in RNA is formaldehyde. So formaldehyde reacts with bases of RNA, forming covalent bonds that prevent the formation of secondary structures. This is crucial for techniques like RNA gel electrophoresis, where proper denaturation is important for accurate separation of RNA fragments based on the size. So MOPS is not used, calcium acetate is not used, glycerol is not used. MOPS is a buffer, so it is commonly used in RNA solution but not for secondary structure. It does not denature RNA secondary structure. It is used to maintain pH of the solution during RNA manipulation. Then calcium acetate is used to precipitate the nucleic acids. While it might affect RNA, but it does not denature the secondary structure and it is not generally used for this. Then glycerol is a viscous liquid which is added to the sample to increase the density. Like I said, when you load it on the gel, you need it to go and sit in the bottom of the well so that it doesn't diffuse in the buffer. So glycerol is used for that purpose, so that it helps them let sink into the loading well during electrophoresis. It plays no role in denaturing RNA secondary structure. So next is ETBR. So RNA in an agarose gel electrophoresis can be visualized by staining with ethidium bromide, also called as ETBR. So why ETBR is used to visualize RNA is because it is flat, it is aromatic molecule, and it intercalates between the stacked base pairs of the nucleic acid. So you have your secondary structure of your RNA and you have your hydrogen base pairs. So what your ETBR does is it goes and sits in between those base pairs so that when you put it under UV, it illuminates along showcasing your nucleic acid. So the fluorescent property when bound to nucleic acids, ETBR shows a strong orange-red fluorescence under UV light, that is around 300 to 312 nanometer wavelength. The fluorescence intensity is much greater when ETBR is bound to nucleic acids rather than when free in solution. So when it intercalates, obviously it will have a much better signal when it's freely floating in the buffer or anything. So when it intercalates in the DNA, it will stack along and give you much better fluorescence. So the mechanism of staining is in agarose gel electrophoresis, RNA samples are separated based on the band size. So ETBR can be incorporated into the gel before polymerization. So when you make your agarose gel, before it cools down, before it solidifies, you add ETBR in it so that it is evenly mixed in the gel. And when you load your sample, your DNA is running, it will collect your ETBR on the way. So or used to soak the gel. So if you don't add it in the gel, you can also use the buffer and you can soak it in the ETBR containing buffer, which is not generally advised because ETBR is carcinogenic. So it's better use it in a gel which gets solidified and later you can discard it appropriately. So in both methods, ETBR bind to the RNA in the gel, enabling us to visualize it. Other options like trypan blue, as I said, is to cell viability. Vital dye is also for staining the cells, and Evans blue is for dead cells, not nucleic acid. So for nucleic acid, it is ETBR or ethidium bromide. So the advantages of ETBR for RNA visualization is high sensitivity. It can even detect as low as 5 to 10 nanograms of RNA, then works for both denaturing and non-denaturing cells. It is inexpensive and easy to use. Limitation is it is, like I said, it is mutagenic. It can insert into the DNA and cause mutation. Basically, it's intercalating between the DNA. So even if you are exposed to it, it will intercalate into the cells of your DNA, into the DNA of your cells. So gloves and proper disposal methods are mandatory. You can use safer alternatives like Cyber Green 2 or Gel Red, which are less toxic, but these are very expensive. So, depending upon the affordability and price, you can go ahead with your choice. So you can see this is your gel under UV. You can see these are the bands that you can see of your nucleic acids. This is because your nucleic acids have ETBR intercalated between them, which is giving you the fluorescence. So this is your genomic DNA. This is your smear. Your mRNA comes as a smear, and this is your 28S rRNA, and this is your 18S rRNA, and you might also have small RNAs here. So your illumination, your visualization is because of ETBR. Next is ammonium acetate. So you use ammonium acetate for destaining the RNA gel after agarose electrophoresis, gel electrophoresis. Then ammonium acetate is commonly used to destain the RNA gel after staining it with ETBR. It helps remove excess stain from the gel, allowing it for clear visualization. Sometimes if you have added more ETBR, your band might be so big and it might be so uh, like a blob. So if you want to use it for publication, you cannot use it. You need nice, neat, crisp, clear bands like this. So you can also remove your excess ETBR by using ammonium acetate, allowing for clear visualization. The DVC treated water, other options were DVC treated water, glycerol mops. These are not used for destaining. DPC, like we discussed, just to prevent RNA degradation. Glycerol is to help in increasing the density. MOPS is for pH. So I think this is done. So does anybody have any doubt in these topics from here after the break? Please let me know. So if there are no doubts, we can stop the class, stop the session. I'll wait for 5 minutes. If anybody has any doubts, you can ask me, or others who do not have any doubt can exit the call. That is the end of this session, everybody. Thank you. See you next week. I'll just wait for 5 minutes just in case anybody has any doubt.
Ma'am, where can I get the lecture and PDF? Which channel? Ma'am, on which channel we can get the PDF and lecture?
So there is, you get your mails, right? I don't know how you usually get it, but we usually share a YouTube link and a drive link for the PPTs. You should be receiving it. If not, I will share you the link of the channel in the chat box here. Just give me a moment. So yeah, so this is the channel. So you can see the videos here of week 1 and week 2 already given here. I will be uploading today's video. So I think you can just screenshot this, or I don't know how to just share this. Okay, you can copy this link. You can go to this YouTube channel. You will have videos over there. But anyways, the link will be shared. I think you have a group where you get all these links and PPTs. If anybody knows like how you get these access, you can share it in the chat box. Because as far as I know, it will be shared to everybody who is present in the group.
Till now, we are only using the mail. Thank you,
You are
ma'am.
only using what?
Ma'am, we can get the link only through the mail. We are not added in any such groups, I think.
So you did get it right the first week and
Yes,
second
yes,
week.
ma'am.
The same way you will get
Ma'am,
this
through
also.
you. Okay, ma'am.
So
Thank you.
once I'm done with this, I will upload it maybe by tonight or mostly by tomorrow. So you can copy this link so you can just directly access and check the channel if it has been uploaded or not. Okay?
Yes, ma'am. Thank you.
Yeah. Anybody has any other doubts? Like I said, you can also mail to me in this, if even at later any point of time, if you have any doubt, you can mail it to me. I can answer it over the mail also. Okay, so I think we can end the session now. Okay, bye everybody.